Hi, everyone. Welcome and happy Wednesday. This is Caroline with Fiveable, as always. And tonight we're going to be talking about DNA replication. Super important. As with everything that we talk about, a lot of things are going to be building off of this concept. So make sure that you tell your friends they should have been here, and definitely feel free to obviously rewatch, watch the replays, etc. But here's your link. Ooh, we got a big crowd tonight. I'm excited. Perfect. Great. Well, if you're just joining us or you haven't joined before, as I said, I'm Caroline. I teach AP Biology in the city of Chicago. And we have talked about a lot so far this year. So if you are just joining us, definitely make sure you spend some good time looking at the replays of all the other sessions that we've had, because bio builds off of itself. And we'll certainly be doing that. If you are just joining us, you're joining us at a great time, because we're kind of moving into, if there were such thing as units, we're kind of moving into the discussion of genes and proteins and genetics, and then we'll move into the human body. It's truly all of my favorite stuff. But we've talked a lot in the past about evolution. We've talked a lot about cell signaling. We have talked about cells as a whole, the plasma membrane, cellular energy, all sorts of stuff like that. So definitely check it out if you haven't joined us before, but I'm very glad that you're here tonight. So as I said, tonight we're talking about DNA replication. I have a couple really helpful tools that I'll use to help explain this. Students oftentimes struggle with the directionality of DNA, meaning like 3' to 5', 5' to 3', etc. So those types of things we will hopefully get an answer to. I have a really great trick with my coach that usually helps to make a lot of sense of 5' business. So stay tuned. And Legos. I have Legos tonight also. Lots of good things. All right. So again, you have the PowerPoint. I'm going to share us on it now. Okay. DNA replication. So we're going to start by taking a look at DNA. I know we mentioned the structure of DNA, and hopefully you guys know the function of DNA, right? It's our genetic code. It makes us who we are. The proteins are the actual workers, right? So when we start talking about transcription and translation shortly here, not next week because Thanksgiving, but the week after, we can certainly talk about that. But yeah, DNA makes us who we are, and we're going to talk about how it's replicated. But as I said, we're going to start out by talking about what is our DNA, what does it look like, why does it look that way, etc. We talked a little bit about this with macromolecules, but our last review is essential. Okay, so a couple of things here, and these are pretty basic, and I'm sure everyone is familiar with this, right? But DNA is made up of these little monomers called nucleotides. Okay, and there's 3 parts to a nucleotide. There's the actual nitrogenous base, which is what we usually think of when we think of DNA. Our C's, our A's, our T's, and our G's, cytosine, adenine, guanine, and thymine, right? But we also have a really important sugar and a phosphate group, okay? In DNA, that sugar is deoxyribose. If you were with us when we talked about macromolecules, anything that ends in ose is a sugar: sucrose, glucose, fructose, right? So deoxyribose is a sugar, and it's where deoxyribonucleic acid, the full name of DNA, comes from, because there's a deoxyribose sugar there. When we talk about RNA, like when we're talking about transcription and translation, we'll talk about ribose, which is the sugar for RNA, right? Which is why it's ribonucleic acid. But this one is missing an oxygen, deoxy, so it's deoxyribose. We have deoxyribose sugar, we have a phosphate group, and we have a nitrogenous base. Okay, those are the 3 components, and I'm going to show you what these bases actually look like on the next slide, because that's kind of important as well. Okay, hopefully you also already know this, but C's, cytosines, and guanines always pair together 10 times out of 10, right? So you see cytosine, guanine here, guanine, cytosine here, and you'll notice that there's 3 little dotted lines in between them. Those represent hydrogen bonds. Okay, so if you haven't had chemistry before, or if you had and have forgotten, hydrogen bonds form between hydrogen, surprisingly, and either nitrogen, oxygen, or fluorines. So we call them FON, F-O-N, right? And in this case, this hydrogen is pairing with one of those molecules and it's making a bond. In this case, it's making a triple bond. We have 3 bonds here that are holding together our cytosines and our guanines, okay? This is in juxtaposition to the adenine and the thymines, which also always pair together. So we have adenine, thymine here, thymine, adenine here, and those are only held together by 2 hydrogen bonds. Why? Well, it all has to do with the structure, which we're going to see a little bit on the next page, but there's just something missing from adenine that guanine has that allows it to make that 3rd bond. Is it super, super essential that you remember the difference between which ones have a triple bond and which ones have a double bond? Not really. If you continue on to take different biochemistry classes in college or even microbiology classes in college, I was a microbiology major, and so we talked about adenine and thymine having weaker bonds pretty frequently because bacteria actually use that to their advantage. So there is a time and a place where that may come in handy, but I haven't seen it appear on an AP exam, okay? But still, kind of important to know. I always like to tell you like why you're seeing the things that you are, and someone would probably ask why are there 3 lines versus 2 anyway, so here's your rationale. All right, these are what the bases actually look like. The big thing that I'll point out, and the reason that I wanted to show you this, is that cytosine and thymine look really, really similar, minus there's 1 methyl group over on thymine that cytosine does not have. And we have, like, our amines are set up a little bit differently. All right, give me 1 second. My dog is being the butt. Apologies. I usually run sessions at school, in which case she is not a problem. Anyway, so our cytosines and our thymines look very similar. We call these 2 bases pyrimidines, and I'll type that into the chat box. Pyrimidines, okay? And so they share that very similar, smaller structure. This is in juxtaposition to guanine and adenine, which have this larger, bulkier structure. Adenine and guanine are purines. Okay, so I like to remember this by remembering that ag, like A and G, as in agriculture, ag is pure, right? Purine, right? So if you think about agriculture, planting and farming, those are very pure things. So I think that ag is pure, and then I remember that cytosine and thymine, therefore, must be the other thing, okay? The reason I'm highlighting this and showing you kind of the structure of these bases is because it provides even more of a justification to why C's and G's versus A's and T, and A's and T's, I should say, pair together, right? Because if we had 2 bulky bases pairing together, like if A and G were capable of pairing, or C and T, we would have really lumpy, bumpy DNA, right? We'd have some big portions of DNA, some small portions, some big portions, some small portions, etc. It would look a mess, right? But because we have 1 purine and 1 pyrimidine pairing in every single rung of our ladder, that is DNA, we actually have a lot of consistency in our shape, and our DNA is the same width throughout, okay? So that is just an important kind of part to remember, and that's what's going to make these, really nice even bases that are then going to twist up into our double helix. Okay, so hopefully this is all review. It should be from like basic biology, but again, if we don't have a strong foundation, our house is going to crumble. Okay, so I want to make sure you guys feel good about that. I should have also mentioned here, this is a sugar-phosphate bond, right? Sugar-phosphate backbone is what they call this because sugar-phosphate binding. Very straightforward. These bonds are really, really strong, these phosphate bonds, much, much stronger than the hydrogen bonds that these bases have between one another. And that's really important as well because every time we go to replicate DNA or every time we go to transcribe DNA in order to make protein, we're cutting apart these hydrogen bonds. We don't want our DNA backbone to spontaneously break apart, right? But we do need to be able to frequently and successfully cut through all of these hydrogen bonds in order to replicate or transcribe, okay? So there's actually a method to this, as with everything in the human body, which is why I love it so much with the amount of bonds that we have. Okay? All right. Cool. So this is a little bit more complicated. And for the most part, what I've found is that people who haven't taken AP, right, which is all of you, because it's your first time, but have just taken freshman bio, or intro bio, I should say, have not learned about the fact that DNA actually has a direction to it. Okay, so here's another picture of a nucleotide. Let me get my little obnoxious pointer up. And if you've taken any kind of sort of like organic chemistry, one, bless you, because organic chemistry is very challenging and it was not my forte in college. But you'll learn about the labeling of carbons in structures, right? So organic chemistry, chemistry in general, really revolves around carbon molecules. They're the most important molecule that we have, most important atom that we have. And so what scientists do, is they will label them in a particular order, right? Usually clockwise. And that allows us to address those carbons. And we can be like, oh yeah, carbon 3 or 3' is what we call this. That's what that little apostrophe is, prime. 3' is the one that has the hydroxyl group on it. And then you're like, oh yeah, this one has the hydroxyl group on it. We all agree, it's a 3' carbon, the end. Pretty straightforward. Okay, so these are labeled because each one of these little junctures that you see here is a carbon. They usually don't write it because it's just assumed. Okay, so we have 1, 2, 3, 4, 5 carbons in this structure. The ones that we care about are the 5' carbon and the 3' carbon. Okay, and again, if you've been reading your textbook or listening to your teacher or wherever you are in your units, then you've probably heard about the 3' to 5', 5' to 3' directionality of DNA. This is what they're actually talking about. They're talking about the 5' versus the 3' carbon. Okay, so a couple things with this. When we are stacking nucleotides, right, which is what DNA is, it's just a bunch of nucleotides put together, we will stack so that 5' at the top and 3' at the bottom fits in with 5' at the top and 3' at the bottom of the next space, and so on and so forth. And this is where my Legos come into play. Okay, so Legos, we all know them, we all love them, right? The building blocks of our youth. So sometimes they're so hard to get apart too. So if we use Legos as our example here, you will notice that our 5' carbon has a phosphate group off the top, right? That big PO4 balloon is what it looks like in the image. Okay, so I'm gonna actually make myself full screen real quick. Whoop. There we go, that was easy. So if I can get these Legos apart, this was also the struggle of childhood, right? Getting Legos apart. Okay, you'll notice that obviously all Legos have a bumpy side and they have a smooth side. Okay, we can think of the bumpy side of Legos as like the 5' end, because remember, if you look back at the PowerPoint slide, or if you're following along on the PowerPoint slide, the 5' end is the one that has the phosphate group. Okay, so 5' end, nice and bumpy, 3' end, nice and smooth, right? You're not going to put two 5' ends together and build any structures anytime soon. You're not going to put two 3' structures together and build any structures anytime soon. You're going to stack them, right, so that you have 5', 3' binding with 5', then 3', and so on and so forth. So as we're stacking, we're layering those 5' and 3' ends over and over again, okay, until we have a more complete structure. And then we could look at this, if this was like a string of DNA, and we would say, yep, this is the 5' end, and this is the 3' end. Obviously, there are other 3' and 5' ends embedded in this strand, okay? But the topmost carbon and the bottommost carbon are the ones that we're going to look at. So the topmost carbon here is this bumpy 5' end, and the bottommost carbon is the smooth 3' end, right? And so we would say that this is 5', this is 3'. If we flipped it, it would be the reverse, okay? And again, this is all based on the carbons that we're counting, okay? The 5' and the 3', and looking at which one is at the top. So if we go back into our PowerPoint, okay, we can see then when we bind these two together, they're gonna form a phosphate bond here. That's part of the sugar-phosphate backbone, right? And we have the 5' sticking up at the most top part with that big old phosphate balloon. And we have the 3' kind of flat end at the bottom down here. And you can see this is labeled for us 5', 3'. Okay? Any questions about that? Because it's a little bit confusing. You feel pretty good? Cool. Legos help. Legos help a lot. I think I could explain, I should challenge myself. I think I could explain almost every biological concept with Legos, but maybe not. Okay, cool. So we're going to add a couple layers to this then in true AP fashion. Just when you get comfortable, right? We add like 10 more layers. That's what we like to do as teachers. So I'm sure you've heard this before. If not, it's very simple to understand. But DNA is antiparallel, okay? So what that means is where one strand, so if we look at this leftmost strand, okay, where one strand has 5' carbon, the bumpy Lego at the top, and 3' flat Lego at the bottom, the strand that it's attached to is going to go in the opposite direction, okay? So the 3' end would be at the top and the 5' end would be at the bottom, okay? That's just how it fits together. That's how the bonds form. Sometimes I show my students, and this is like kind of dorky, and I'm going to go full screen again because why not? I'll be like, okay, we have a 5' end. That's our hand. It has 5 fingers, and then the 3' end is our smooth elbow. Okay, and we can't have two 5' ends in the same place because they'll intersect one another. They'll bump into one another, and it will cause problems. So what we got to do is we have to flip it. So we have one 5' and one at one end, one 5' at the other end. And then our smooth 3' elbows are also going to be at opposite ends. Okay, and then now we have a fun handshake. So you can go show your friends. It'll be really, people will think you're really cool, right? But 5' end at one end, 5' end at the other, and we call this antiparallel. Obviously, there's still parallel lines, but they're running in the reverse directions. Okay? All right. Any questions about that? I saw it looked like a question appeared and then disappeared. Okay? But that's all that means. 5' end at one end, 5' end at the other. Full handshake. Never forget it. Okay, cool. So the plot thickens. That's why I keep asking you if you have questions, because we're going to keep building onto this. So once we talk about actually DNA replication, which we haven't talked about yet, then we're going to add a couple more layers. Super important foundation here. 5' to 3', antiparallel will make it 3' to 5' on the other side. And it's just the directionality based on the carbons that are present in the nucleotide. Okay, cool. Here we go. Getting my PowerPoint back up for you. All right. So the replication process itself, we should probably start talking about that because that's what our session is about tonight. And so the replication process has been boiled down, and scientists did a lot of different experiments to figure this out, as always. But it has been boiled down to being described as semi-conservative, which is something that you may have heard before as well. Okay, so in order to explain this, I'm actually going to show you 3 different potential models, and I'll explain the semi-conservative one, obviously, in the most detail because that's how DNA actually replicates. But basically, so scientists at one point thought, maybe DNA just like completely replicates itself, right? So the parent is pink here, and then we make a whole new strand for both strands after a replication cycle. And then we continue on like this. So this one remains fully parental DNA, this one remains fully new DNA, and that's how it replicates. There was also thoughts that perhaps we took segments of parental DNA and combined it with segments of new DNA and had some kind of like mosaic pattern like you see down here. That was called the dispersive model. And these were disproved, and you probably already know this because you probably know a little bit about DNA replication, and overtaken by the semi-conservative model. Okay, and what that means is we're conserving half of the DNA from the parent, and we're making the other half from a brand new strand. So this parental DNA will split, which is what you're probably very familiar with, right? It will split open and will replicate one side, and that will become a whole new DNA molecule that's half parental DNA and half new DNA. And then the other end will get replicated as well, and we'll have another strand of DNA that's half parental and half new. And you can see that as this mix of pink and blue in this slide. Okay, so this is the way that this happens. And this is going to be really, really important in how we minimize errors in our replication process because we're using the parent as a template. So knowing that only adenines and thymines can pair together and cytosines and guanines can pair together, and knowing that we're using a template this is pretty foolproof when we go to replicate our DNA. Oh, also, this is a fun thing. I can't take credit for it. Another teacher at our school came up with it or found it somewhere. But if you ever struggle with the fact that A's and T's pair together and C's and G's pair together, you can think apples go in the trees, cars go in the garage. So apples in the trees, adenine with thymine, cars go in the garage, cytosine pairs with guanine. Okay, so if you've ever struggled with that, there's your little acronym to help you out, or analogy. Yeah, whatever it is. Okay. All right. So we're gonna dive deeper into this semi-conservative model then, right, where we're actually splitting that parental DNA and we're adding a new strand in order to replicate this. If you're wondering, when in the world does DNA replication take place? It's all the time, anytime that your cells are replicating, right? So sometimes students get a little bit confused with DNA replication and then transcription and translation, which are going to be wholly different processes. But DNA replicates anytime your cells are splitting. So like your hair cells, the cells underneath your skin, right, that are making these dead outer hair cells, those are going to be replicating all the time. So they're going to be duplicating your DNA before they replicate. Same with cells inside of your intestinal tract. Your skin cells are constantly replicating. So some cells don't replicate all the time, but those cells replicate quite frequently. And so they'll be doing DNA replication all the time using the semi-conservative model. Okay. So what actually happens here, right? So I've shown you about DNA. We've talked about its directionality, which is going to come into play. And we've talked about how this happens in a semi-conservative manner. So now we're going to start talking about the key players, okay? So the first thing that happens in order for us to replicate DNA, first of all, I should have added a picture of that. I'll pop one in a little bit. But we start replicating at what we call the origin of replication, which makes a lot of sense because origin is the starting point. Replication is what we're doing here. So we basically form these little bubbles in the DNA. Let me get a good picture up really quick. Sorry, guys, I apologize. I want to make sure you get a good image here to work with, so you understand what in the world I'm talking about. Okay. Ta-da! Origins. Okay, so we'll look at the eukaryotic side because that's what we're really talking about here. But there'll be a couple different origins of replication, a lot of different origins of replication on a single strand, right? And that's where the DNA will start to split in both directions. So we'll have replication forks going in either direction away from this origin. And then we'll start replicating in both directions until these bubbles will eventually join one another, right? Because this one will be moving in this direction. This one will be moving in this direction. At one point, they will intersect, and then we'll have two new strands, one on the top and one on the bottom. Same with here, coming together, two new strands, and whatever other origins of replication exist in this strand. Okay, until we finally have those two complete strands. So it's not just like we open it at one end and we get everyone confused. And we replicate both sides just all the way through. That would take forever. We would all be dead. Okay, so instead we have lots of different origins where we're starting at the same time and moving towards one another in order to expedite the process. Okay, so that's what we're seeing here. And the first enzyme that we'll talk about is an enzyme called helicase. Okay, so friendly reminder that anything that ends in ase is an enzyme. Anything that ends in ose, which we talked about earlier, is a sugar. Anything that ends in ase is an enzyme. And this is the enzyme that breaks apart the helix, right? Which is why it's called helicase. Okay, so basically what helicase does is it breaks those hydrogen bonds that we were talking about, those relatively weak bonds. It comes through and it snips all of them. Okay, so it's snipping these guys over here. It's snipping these guys over here. There's another enzyme, topoisomerase, which I don't have on this diagram, but I do have it on one of the other slides. Topoisomerase is going to help to break some of this tension here. So it'll make little micro breaks in the sugar-phosphate backbone so that we don't have like all chaos break loose. I wish I had a string. But if you've ever tried to like unravel a string or a piece of yarn and you keep unraveling it, it'll start to get really bunchy up at the top as all the tension kind of builds. And that's basically what would happen to our DNA if we didn't have topoisomerase making little micro tears in it. Okay, so helicase is breaking these bonds for us because obviously we need to break them in order to start replicating the parental strands, but we wouldn't be able to do that without the help of single-stranded binding proteins, which are these guys over here, which basically just grab the exposed DNA and keep them apart from one another, okay? So that's really, really important as well because these guys want to be bound to one another, right? That's why they are bound 99% of the time. So once they're ripped open, they're going to try to bind back together. So we use these single-stranded binding proteins in order to hold these strands apart so that we can actually get DNA polymerase in there to replicate. Okay, so super important proteins there. Helicase, gotta know it. Single-stranded binding proteins, abbreviated to SSBP. Bailey, great question. Not all of these models happen from the previous slide. Only the semi-conservative model happens. The reason I have the other ones up there is because those were ones that scientists once believed were the models for DNA replication, and we have thus disproved it. But it's kind of hard to understand the semi-conservative model without seeing that other models existed. Also, I should add that scientists didn't even know that DNA was our genetic material 100 years ago, right? That's nuts. 100 years ago is not that long ago, but scientists for a very, very long time, they knew that we had genetic material, right? But they thought it was actually protein because protein has more bases, right? Bases has 20 amino acids. And it has a more complex structure. So they were like, oh yeah, that's definitely what our genetic material is coded in. Obviously, we know that's not to be true now. You've probably known for your entire life that DNA is our genetic material. But that was only a realization that scientists came to like 80 years ago. So progress. Anyway, I digress. So helicase is breaking those bonds. Single-stranded binding proteins are keeping those strands apart from one another, and they're going to lay the foundation for everybody else. So let's keep cracking. All right. So up next is this enzyme, which is kind of like an orange-looking thing, an orange blob, and this enzyme is DNA polymerase. Okay. So I know it can seem kind of confusing to memorize all these different enzymes, but their names make a whole lot of sense. So with helicase, we're breaking apart the helix, right? So helicase makes sense. With DNA polymerase, it's the enzyme that makes the polymer DNA. Remember that a polymer is just a grouping of monomers, right? So if a Lego is a monomer, then my beautiful Lego tower is the polymer, okay? And so it's making this polymer. So DNA polymerase is the thing that scoots along the exposed DNA and says, oh, this is an A, I'm going to pair a thymine here. Oh, this is a G, I'm going to pair a cytosine here, right? And it runs along the exposed open parental DNA and places the corresponding bases onto the parental DNA, making our new strand, okay? All right, here's the thing though. That 3' to 5', 5' to 3' thing is coming back, okay? And it's because DNA polymerase can only work in one direction. It can only bind in one direction. It can only move in one direction, okay? The good news is it reads the parental DNA just like we would read a book, right? From 3', you can see down here, we're moving in this direction, right? Right? So from 3' to 5'. Okay, so I always tell my students, you shouldn't really have to memorize this. If you can remember how you would read a book from page 3 to page 5, not from page 5 to page 3, right, then you can remember that DNA polymerase moves from 3' to 5'. Always, no exceptions. This is going to come in handy in a couple slides when we talk about my favorite word in bio, which is Okazaki Fragments, okay? And this is the reason why those things exist. This is the reason why we have leading and lagging strands, if you've heard of them before. If you haven't, stay tuned because we're going to talk about them in a bit. Okay? But again, DNA polymerase is unidirectional. It only moves in one direction, and it moves reading the parental DNA from 3' to 5'. What that means is the new strand of DNA, right, the new strand is going to be synthesized or created from 5' to 3'. because DNA is antiparallel, right? So while it's reading from 3 to 5, it is creating or synthesizing from 5 to 3. But if you can remember how you read a book and you remember that DNA is antiparallel, then that should kind of fill in itself naturally. Okay, so and again, we're seeing this new strand being made, and it's being made from 5' to 3' as DNA polymerase moves along the exposed bases of the parental DNA strand. Okay? Looks like I have a question. Ooh, okay, great questions. Aditi, is there a specific sequence that initiates the origin of replication, or is it just random? Great question. There is going to be specific sequences that start the origin of replication, okay? And basically, there's another enzyme that we're going to talk about in a second called primase that's going to put down the primer so the DNA polymerase knows where to start. Great question. Stay tuned. If I don't answer it, please re-ask it, but we should answer it in a couple slides. Kimberly, this is where antiparallel comes in. So DNA in its entirety, whether it's being replicated or just sitting in your cells, is always antiparallel. Again, because if there's two 5' ends, they're going to crowd one another. So we have to go 5' to 3'. This is where we're seeing that play out again, because while we're reading from 3' to 5', we are creating or synthesizing DNA from 5' to 3'. Vera, I see your question. I'm going to save it until the very end to see if we have time. It's great, but I want to make sure we get through everything that we are supposed to. And then I would love to talk to you about prokaryotic versus eukaryotic DNA, because as a microbiology major, most of the DNA that I studied was actually prokaryotic. Okay, cool. Any other questions about the 3' to 5' business? Okay, my code example is coming up. So if you've been waiting, thank you for waiting. It will be here shortly. Okay. So I'm going to give you 1 second to take a look at this while I go grab my coat, and then I promise I will make sense of it all, because it looks like a mess, but it's actually relatively straightforward. Okay, so take a look at this. I'm going to go grab my coat. I'll be right back. Okay, I'm back with my coat. So here we go. All of your questions shall be answered. So this is where we get leading and lagging strands from. This is where we get my favorite word, Okazaki Fragments from, and I can explain to you why. Okay, so let me first orient you to the diagram itself. So if you follow my pointer here, right, this purplish, darker blue is our parental DNA. We can see that it's connected right here. We can see that it is moving in 2 different directions because DNA is antiparallel. So while our top strand has the 3' on the left-hand side of the slide and a 5' on the right-hand side of the slide, our bottom DNA has 3' on the right and 5' on the left, right? Again, antiparallel strands, this is going to come in to play in a big way here, okay? Our replication fork, which is what we call the area in which helicase is unwinding or unzipping, hence the coat analogy shortly here, is moving towards the left of your screen. So helicase is coming through. These little black balls are single-stranded binding proteins, okay? But helicase is coming through and breaking these bonds for us. It's moving towards the left of the screen, right? And we're opening up more bases that need to be replicated, okay? Now, remember, DNA polymerase can only move from 3' to 5', okay? So this bottom strand has it made in the shade, okay? Everything is good for this bottom strand because 3' is over here on the right and 5' is over here on the left. And so DNA polymerase will move towards the left of the screen, okay? Filling in bases, making a complementary strand that's this light blue shade that you see here. And it can continue to move in this direction, and it's moving in the same direction as the replication fork. So as helicase opens up more DNA base pairs, DNA polymerase will follow it and continue to add more DNA base pairs until it runs into another replication fork, right? So it can move in the same direction it's following that helicase forever. As helicase moves to the left, DNA polymerase will follow, reading from 3' to 5'. Okay? Unfortunately, the same is not true for our top strand, okay? Because, and students get this confused all the time. They'll be like, oh, the lagging strand goes in the opposite direction. It goes from 5' to 3'. It doesn't. The reason the lagging strand is a lagging strand is because it still has to go from 3' to 5', right? So again, DNA polymerase can only read from 3' to 5', okay? This becomes a problem for this top strand because guess what? 3' is over here, 5' is over here. It's working towards the right, right? It is working towards the right, which is away from the growing replication fork, okay? So if you follow my pointer here, and then I'll show you my code example, and then I will certainly answer any questions that you have, right? As we're moving here, right, away from the replication fork, replicating the DNA from 3' to 5', building DNA from 5' to 3', right, antiparallel, the replication fork is moving in the opposite direction, right? So we are truly working in the opposite direction. So if we're starting at the same point, right, and DNA polymerase is moving towards my right, and the replication fork is moving towards my left, right, all of a sudden, there's this whole gap that needs to be filled in because this guy went and unzipped a whole bunch of more base pairs while I was busy moving in the opposite direction. So then DNA polymerase has to hop back over here and fill in the gaps. But as it's filling in the gaps, helicase is is opening up more gaps, okay? So I'm going to show you what that looks like with my coat really quick because that's going to help to explain why we're kind of like moving against the stream. We're moving in the opposite direction of the replication fork, and that's going to cause us not really a problem because this happens to half of our DNA and we're certainly just fine, but it just causes a minor complication, okay? So Kimberly, this is not, it doesn't have any sort of bad effect. It's, again, it happens to half of our DNA. It's just the nature of DNA replication. It doesn't mean that we have any more mutations or anything like that. That ligase that you see on your screen, that's that bright red, looks like a kind of like a blood droplet, I guess. That's going to seal together the 3' and the 5' ends of these little fragments. And so it will look just like DNA. If we looked at DNA underneath an electron scanning microscope, we would not know which one was created via a leading strand and a lagging strand because it's all intertwined and it ends up perfect in the end, right? Thank you, human body, for being so wonderful. It's just kind of the nature of how the replication needs to occur, okay? All right, so take a look at my code example because DNA is really just like a zipper, right? And helicase is unzipping, right? Unzipping the genes in order for us to have a reaction. So it's coat time. And if you guys don't know, Chicago is like 20 degrees right now, so I need my hefty winter coat. Maybe it's a little warmer where you are, maybe not. I think it's kind of cold everywhere right now. But I'll zip up my coat. I'm going to make myself full screen and explain, okay? Okay, you can see my lovely bookshelf, which is just brimming with AP Bio books. Okay, so put on my mask. I know it's probably not the easiest to see, but it'll still get the idea across. Okay, so my zipper is the DNA, right? And as I'm unzipping, right, this would be helicase, right, where my hand is, right here. This is helicase, it's unzipping, right, and we're exposing new bases to be replicated, okay? Now remember, DNA is antiparallel, okay? So I'm gonna start, I'm just arbitrarily naming the different parts of my zippers, okay? But let's say the top part over here on my left, I think it's your right, is the 3' end of DNA, and this is the 5' end, okay? We're gonna start over here on your right side, right? Because this is the 3' end, and this is going to become my leading strand because I'm going to be moving in the same direction as helicase, as my zipper, okay? So as I start up here, this is my 3' end. I'm laying down new DNA, okay? And as I'm laying down new DNA and the zipper opens up more, you'll notice that I can just follow it all the way down until I get to the end of the coat. No problem, right? We're moving in the same direction as the replication fork. So as my DNA polymerase moves down, it's following the helicase and we're able to replicate no problem. As it opens more bases, the DNA polymerase follows. That's our leading strand. Our lagging strand, on the other hand, has to be the other half, right? Because if this is 5', my 3' is down here all the way at the bottom. And so we're going to be moving from 3' to 5', which is going to be up towards my head, as opposed to down towards my toes, like was over here, okay? So as I'm unzipping, I'm going to be working upstream, right? I'm going to be working in the opposite direction, okay? So we're going to use these little buttons as markers, okay? So that you can see the progress being made, okay? So as I said, we're going to have to be moving up towards my head, okay? Because that's 3' to 5', and that's the only way that DNA polymerase can move, okay? So here I am, little DNA polymerase, right? Here's helicase. I'm starting at this first button right here, and I'm going to start replicating DNA. So I'm going to replicate. As I'm replicating, though, what's happening? Helicase just opened up a whole stretch of DNA that now needs to be replicated. I replicated from here, this first button, up to the top, which is lovely, right? But now there's a whole new section of DNA that's been opened up while I was working that I'm going to need to hop down and replicate. Okay, so here's my new starting point, right? I've made it to this new button, right, as I've unzipped. And as I'm moving up, because now this whole area from button to button is exposed, as I'm moving upwards, right, DNA polymerase is unzipping more, okay? So it's all well and good that I just filled in this segment, but now there's a whole new segment, a whole bottom of my coat that needs to be filled in. Okay, so DNA polymerase would have to hop down to the bottom and fill back up to where it left off. Okay, so in my coat analogy, we had 3 big segments. We had the bottom segment, a middle segment, and a top segment. But the reason that it was so broken up is because we're working against the direction of the replication fork. So as DNA polymerase is moving up towards my head, helicase is moving down towards my toes, right? And it's opening up more bases. Aditi, that's a great question. How does DNA polymerase know to move backwards? So in reality, there's lots of DNA polymerases that are probably working concurrently at the same time. Basically what happens is these enzymes are just like kind of moving around. So when that first one is done, it'll just kind of be moving around in free space until it bumps into a primer and knows to start replicating again. So it's kind of just like a chemical reaction that happens. So there's lots of DNA polymerases around. They're kind of spontaneously moving and finding those primers, and then they're starting. Really great question, okay? So that might need a rewatch. As with anything, rewatching things is really important. But if you are struggling with leading and lagging strands, coat example is a really good one. And you can also try it on yourself, right? Get your coat out, see what happens when you move your zipper down and your hand up, right? You're not going to be able to replicate the entire zipper in one fell swoop like you would for the leading strand where you're moving in the same direction as the replication fork, okay? If you feel ever so slightly better after that example, I am thankful. It's a good one. Bailey, yes, both strands are being replicated concurrently at the same time. And remember that there's also lots of different origins of replication, and we're moving in both directions at each one. So it's actually even more complex that we're moving in 2 directions at lots of different points all at the same time, which is how DNA replication happens so quickly. We can replicate all of our bases in like 6 hours, in like a few hours, which is pretty crazy. An E. coli bacteria has 5 million bases, and it can replicate all of its DNA in about an hour. So our DNA polymerase actually puts down 1,000 bases in 1 second, one single second, 1,000 bases in a second, which is like faster than we can even fathom in our brains. Kara, the lagging strand is not a mutation. It happens all the time. It happens half the time, right? Like there's always going to be a lagging strand and a leading strand. And the reason that it happens is because DNA polymerase can only read from 3' to 5', and DNA is antiparallel, okay? So there's always going to be one strand that's moving with and one strand that's moving against the replication fork because they go in the opposite direction. And DNA polymerase can only read from 3 to 5, Okay, so DNA polymerase is always going to be working in opposite directions on 2 strands of DNA because they're antiparallel. So that's why it occurs. But not a mutation, just a regular part of replication. Just a crazy fun thing. So those little fragments that we were building, those are called Okazaki fragments, right? Because the dude that discovered them had the last name of Okazaki, okay? And so those fragments are going to get glued together by that enzyme ligase. So if I pop back into our PowerPoint real quick, that was the big part of tonight. So if you feel decent about that, you're pretty good, okay? So ligase is the enzyme that ligates, which is like a fancy word for glues, but it will seal together these Okazaki fragments. It will seal together the sugar and phosphate bond so that these Okazaki fragments are no longer fragments, and we have nice smooth DNA, just like we do in the leading strand, all connected. Okay, so thank you, ligase. It's going to glue all those together for us and make it one nice long strand. Okay. There are a few more things that we need to be familiar with. So this was going back to the question about how does, where does it start? Is there a special region that it starts in? And that's going to be even more specific, the regions of starting and stopping, when we actually talk about transcription and translation in making proteins based off of certain genes. But in DNA replication, there's also starting points, right? The origins of replication. And those get triggered by primase, which is an enzyme that puts down primers. So again, I know it seems like you have to know a lot of different words, but they all make sense, okay? And so what primase does is it puts down little RNA primers, which are these orange blocks that you see here and here, okay? And it puts down these primers, and that gives DNA polymerase the go-ahead to get started, okay? So DNA polymerase is not actually capable of binding onto these open base pairs like this. And so it needs these primers to be put down like this by primase in order to know where to start and also grab onto the DNA strand, like hop on. It's like a little bead, so it has to hop on like this and start replicating. Okay, so that's the job of primase. And you'll notice that primase put primers in 2 opposite locations here at the 3' end of the parental strand. The 3' end and then 3' end is where the primers go because that's where DNA polymerase will bind and then move down the line. Okay, so that's another important enzyme. And I have a table of all the important enzymes that you should know at the end in case you're like, oh my goodness, this is a lot. I have it organized for you at the end. Okay, but we're truly almost there. Okay, there's another example of this, right? So each of these little segments actually would be started off by an RNA primer. So this orange circle here is primase. That's putting down these little segments of RNA, right? And then we're replicating from those regions because that's where DNA polymerase, technically DNA polymerase 3, which we'll talk about in a little bit, binds and replicates. Okay, pretty straightforward. But again, DNA polymerase cannot bind to just the parental DNA. It needs the little RNA primer put down by primase in order to know where to bind, in order to start. Oops, I should have added this, and apparently I didn't. These primers will then need to be removed, right? Because we don't want RNA in our DNA. We don't want ribose sugar in our DNA. We don't want uracil in our DNA. So DNA polymerase 1, and I'll type this into the box because I didn't put a picture for you. My apologies. DNA polymerase 1 comes in and replaces the RNA primers with DNA. And then ligase fills up the gaps in the sugar-phosphate backbone. Okay. So with primase is always also going to bind to the 3' end because that's where DNA polymerase is going to start, right? It's going to start at the prime end. So that's the direction that primase will move in from 3' to 5', and it just lays down a few bases, not many, just a few, in order for DNA polymerase to bind to that area, okay? All right. Again, these will be replaced by DNA polymerase 1. Don't be super stressed that there's different DNA polymerases. For the most part, you just need to know the basics. I mean, they're both doing the same thing. They're putting down complementary base pairs, but DNA polymerase 3 is the one that's laying the big strands of DNA, like you can see here. And DNA polymerase 1 is like more of a proofreader. So it's going to eliminate those RNA bases and replace them. And it's also going to be our editing tool. So if I flip to the next slide, if you can imagine, right, so as I said, DNA polymerase 3 puts down 1,000 bases in a single second, which is way faster than our brain can even comprehend, okay? If you had to type 1,000 words in a second, right, not in a minute, in a second, you would make a lot of errors, right? So DNA polymerase 3 does make a decent amount of errors, but thankfully, DNA polymerase 1 can catch on to them. So there's a lot of different errors that can be formed, but this goes back to the beginning where I was telling you about the different shapes of the molecules. So let's say on accident, an adenine and a guanine are paired together. That's going to be really, really big because they both are really, really big bases. And DNA polymerase 1, as it's proofreading, will pick up, ooh, that's not right. It will remove the incorrect base, place the correct base, and life is good, right? So it will do that, or if it's too small, or if it's like bound in a weird way, it will do that and proofread any sort of error that might have happened from the fact that DNA polymerase 3 moved really quickly. Okay, so what happens is we go from having an error every 1 in 10,000 base pairs to having an error every 1 in 100 million base pairs. So 1 in 10,000 to 1 in 100 million, all because of DNA polymerase 1 coming and proofreading, right? Which is a public service announcement that you should always proofread your papers before you give them to your teacher, because we can tell if you didn't. So always find your DNA polymerase 1, someone to proofread your paper, to make sure that you didn't type 1,000 words a minute, okay? Kimberly, do all polymerases have different functions? So DNA polymerase 1 and DNA polymerase 3 basically have the same function. It's just that DNA polymerase 1 is proofreading and putting in the base pairs, but it's still pairing up the bases in a complementary fashion. And DNA polymerase 3 is just doing the heavy lifting of actually creating the strand in the first place. A polymerase is just an enzyme that makes a polymer. So when we talk about transcription, we're going to talk about RNA polymerase, and that makes the polymer RNA. It's still going to put down complementary base pairs just like DNA polymerase does, except it's going to put down matching RNA bases instead of DNA, hence the name RNA polymerase. Looks like I have a couple other questions. So Vera, if DNA polymerase 1 is damaged or stopped functioning, we would have pretty mutated DNA. I don't know off the top of my head. Students always ask me the best questions, and I don't know all of the answers to all of them. But that's a really good thing to look up if there's like a disorder that's a mutation in DNA polymerase 1. I would assume that that would be so detrimental that if someone had that, they wouldn't be born. They wouldn't actually survive the birthing process because it's just too severe. There'd be too many mutations. But I don't know that for sure. So that's something I would encourage you to look up. But for sure, there would be tons and tons and tons of mutated DNA if that were the case. Okay. Last slide, guys. 6:53. Thanks for spending your evening with me. Oops, not last slide. I lied to you, but it can be. This is mainly just review, right? So this is all of our enzymes coming together. Okay, so we have primase putting down our primers. We have, so that's what's here, right? So this primase has put down this little RNA primer. We have DNA polymerase, which is moving in this direction and in this direction. Okay, it's kind of hard to see because these are flipped. And then we have a helicase, which is unzipping. We have single-stranded binding proteins keeping these guys apart. And then we have topoisomerase, which I said was kind of like making little microcuts in order to basically flatten out our ladder. Okay, and then we have ligase sealing up our Okazaki fragments right here and here. Okay, so that is that. That's just a little review slide, but we've talked about all of that, so I won't spend too much time on it so that I can have time to answer your questions. But here's the list of enzymes, okay? So DNA polymerase 3 replicates the DNA strand by placing complementary bases down, okay? It's the big heavy lifter. DNA polymerase 1 proofreads DNA and also replaces the RNA primers. It's still placing complementary bases, but it's doing so in the proofreading process and in the priming process, or removing the primers process, I should say. Helicase breaks apart the double helix. It unzips DNA strands by breaking those hydrogen bonds in between them. Primase primes. It puts down RNA primers so that DNA polymerase can bind. Our single-stranded binding proteins, not really an enzyme, but a protein, holds open the separated DNA strands so they do not come back together. Topoisomerases make small cuts in the helix to release tension from unwinding. And then ligases are glue. It glues together the nucleotides that have either been put in place of a primer or have been put in fragments so that the sugar-phosphate backbone is solid and we don't have little breaks in that backbone, because that would be bad. We don't want breaks in our DNA. Okay, so those are the main overarching ideas. Polymerase 3 comes before polymerase 1. Yes, for the most part, it will. Yeah, DNA polymerase 3 is going to do its job first, and then DNA polymerase 1 is going to come back through. Why are they named that way? I do not know. Usually, like when we talked about photosynthesis, and photosystem 2 comes before photosystem 1, it was because of how they discovered them. But it would be hard to believe that DNA polymerase 3 would be discovered last. So maybe the structure of it got discovered last. But yeah, for the most part, DNA polymerase is going to move through first. DNA polymerase 3 is going to move through first, and then DNA polymerase 1 will come by and proofread it and remove any of the RNA primers and replace them with the complementary DNA strands. Any other questions? Cool. So Vera, I'll answer your slightly off-topic question now then. So prokaryotic DNA is circular for a number of reasons. One, it actually allows them to replicate it faster because they can start a couple of origins of replications in an easier fashion. And instead of having kind of like dead zones at the end, they can just kind of zoom around until they meet one another, and then they have those two circles. But two, they just have less genetic information, lots less genetic information than we do. They have 5 million base pairs, and we have in the billions of base pairs, right? So it's just like absolutely nuts how much more DNA we have than them. And if our DNA wasn't organized in chromosomes, meiosis would be a lot harder, which we haven't talked about yet, but we will. And we would have a lot more DNA replication. It would just take too long, right? Because we're replicating like billions of base pairs that are all just in one big section. So by dividing those into those chromosomes, we're able to make them more compact, smaller, right? Because our DNA, if we unraveled it, would be like 2 feet long. So we're allowed to make it more, we're able to make it more compact, we're able to replicate faster, and we're able to take all of our genetic information, which is way more than a prokaryote, and put it into a little cell in a nucleus, right? As opposed to theirs, which is just chilling out in the cytosol. Oh, Aditi, you are 100% correct. So polymerase 3 goes over the DNA once, then polymerase 1 goes over it again to proofread and remove the primers, right? Because again, we don't want RNA in our DNA. It's a different sugar, and it has uracil instead of thymine. So we want that proofreader to come through and make sure that all of that is done. I'm going to go grab my charger real quick. I can stay for an extra minute if people still have questions, but my computer's about to die, and I'll be right back. If you leave before then, have a great Thanksgiving break, and I'll see you the week after. Any other questions? I didn't realize my backpack was right next to me. Sorry, guys. I plugged in my computer right as it was dying, so that's classic. But Kimberly, sorry, yeah, no, it didn't end. My computer just died. But Aditi and Bailey, great answers. So you're absolutely right. 3 comes first, then 1 proofreads. And Bailey, I am so glad to help you out. Let me know if you guys ever need additional help with stuff. My goal is to make things seem a little bit more tangible. AP Bio is really hard. So if you're feeling like this is all a lot, it's because it is. And rewatching this will definitely help make more sense of things. But things like Legos and coats and stuff like that sometimes make all the difference. So keep tuning in. Please tell your friends. It's such a worthy investment, y'all. It's so cheap in comparison. Like you could go out to dinner for a lot more money than you're paying for a year of some extra support. So tell them to join. It's great to have a big crowd, and I really appreciate you guys being here tonight. So have a great rest of your night. Have a great Thanksgiving. If you have more questions, we can chat after the break for sure. And we're just going to build off of this for transcription and translation. So more to come. Rewatch for sure. Oh yes, Vera, I told you I would do that and I haven't yet. So I'll definitely practice. I'll post more practice questions. And I hope you guys have a great night. Bye, y'all.